Welcome back to part 2 of our series on the 100 scientific discoveries that built the modern exam syllabus. In this episode, we journey from the chemical foundations of matter and electromagnetism to the quantum revolution, the discovery of life-saving antibiotics, and the beginning of genetic engineering. Grab your notebook, and let us jump straight into discovery number 21. 21. Antoine Lavoisier and Joseph Priestley, working in the 1770s, identified oxygen as a distinct gas essential for combustion and respiration, overturning older theories about fire. 22. Lavoisier also gave us the law of conservation of mass in 1789, matter is neither created nor destroyed in a chemical reaction, only rearranged. Every balanced chemical equation you write depends on this law. 23. Michael Faraday discovered electromagnetic induction in 1831, proving that a changing magnetic field can generate electric current. Every generator and transformer on Earth runs on this principle. 24. James Clerk Maxwell unified electricity, magnetism, and light into one set of equations in 1865, showing that light itself is an electromagnetic wave. 25. Louis de Broglie proposed in 1924 that particles like electrons also behave like waves, a strange idea called wave-particle duality that sits at the heart of quantum mechanics. 26. Werner Heisenberg's uncertainty principle, from 1927, showed that you cannot simultaneously know a particle's exact position and exact momentum. This is not a limitation of our instruments, it is a fact about reality itself. 27. Otto Hahn and Fritz Strassmann achieved nuclear fission in 1938, splitting a uranium nucleus and releasing enormous energy. This discovery led directly to both nuclear power and nuclear weapons. 28. Niels Bohr, in 1913, proposed that electrons orbit the nucleus in fixed energy levels, rather than spiraling in freely. This model is still the first one taught to school students, even though it was later refined. 29. James Chadwick discovered the neutron in 1932, the neutral particle that, along with protons, makes up the atomic nucleus. 30. Friedrich Banting and Charles Best isolated insulin in 1921, turning diabetes from a near-certain death sentence into a manageable condition. 31. Selman Waksman discovered streptomycin in 1943, the first effective antibiotic against tuberculosis, a disease that had killed millions in India alone. 32. Jonas Salk developed the polio vaccine in 1955 and famously refused to patent it, saying that patenting the sun would be equally absurd. 33. Friedrich Kekulé worked out the ring structure of benzene in 1865, reportedly after dreaming of a snake biting its own tail. True or not, the structure itself transformed organic chemistry. 34. Gregor Mendel, a monk working quietly with pea plants, published the laws of inheritance in 1866. His work on dominant and recessive traits was ignored for decades before scientists realized he had founded modern genetics. 35. Walter Sutton and Theodore Boveri connected Mendel's abstract laws to actual physical chromosomes in the early 1900s, proving that genes sit on chromosomes. 36. Antony van Leeuwenhoek built his own microscopes in the 1670s and became the first person to see bacteria, opening up an entire invisible world. 37. Casimir Funk coined the term vitamin in 1912, after research into diseases caused by deficiency, like beriberi and scurvy, showed that trace nutrients were essential to health. 38. Karl Landsteiner discovered human blood groups in 1901, explaining why some blood transfusions succeeded and others were fatal. This discovery alone is estimated to have saved hundreds of millions of lives. Here is where it gets interesting for exam purposes, because this next stretch is where classical biology hands the baton to modern genetics, the part of the syllabus that keeps expanding every single year. Chapter 3, The Genetic Revolution. 39. Kary Mullis invented the polymerase chain reaction in 1983, a technique that lets scientists copy a tiny fragment of DNA billions of times. Every DNA test you have heard of, from paternity cases to COVID testing, depends on this method. 40. Jennifer Doudna and Emmanuelle Charpentier developed CRISPR gene editing in 2012, essentially molecular scissors that can cut and edit DNA with precision. It won them the Nobel Prize and is now one of the most exam-relevant technologies in biotechnology sections. That concludes Part 2 of our series, Discoveries 21 through 40. In Part 3, we cover the Human Genome Project, Dolly the Sheep, and a dedicated chapter on India's greatest scientific minds including C. V. Raman, S. N. Bose, Subramanian Chandrasekhar, and J. C. Bose. Subscribe and click the bell icon so you don't miss Part 3.